So my name is Chris, and Mark introduced me to the Graython group. I showed up to one meeting last month, and now I'm talking. So, and that coupled with another fact, which is that this audience is almost impossible to prepare a talk for. You kind of just have to go forward. Some of you I know are biology people, some are computational people, some are art, most are all three, honestly. And so if I make assumptions about what a certain word means, and I'm incorrect and it completely baffles you, stick up a hand. If I seem to be making no sense at all, it's probably actually that we don't speak the same language. My background was computer science. I did graduate work in artificial intelligence at the University of Michigan. Back then that meant neural networks, evolutionary computation, and a little bit of vision processing. Some natural language stuff, which humiliatingly enough is now implemented on my cell phone. So I thought it was an interesting bit of research back then, but not so much. Turns out to have a nice algorithmic solution. So in Michigan, I went to work for a defense contractor where we did vision recognition. The question was tank or truck. You've got either a radar image or an infrared image of something. And if you're throwing $1 million munition at a $10,000 vehicle, you're losing. We tried to address that. Unfortunately, we're still losing, and I couldn't talk about it to my friends. So I jumped ship from there into this new hot thing. The human genome was being, done. There's something called bioinformatics. And what I noticed was that funding for science projects in physics was pretty much static. You look at the overall spending from the NIH on physics, it's been static for about 15 years. You look at the overall funding from the NIH on bio, particularly computational techniques attached to bio, and it's just doubling, like every 3 years. It's insane. It's still happening. And so that seemed like a good place to sort of push and be able to talk about my work, maybe save the world, whatever. Went to the University of Minnesota, joined this insanely long-named group called the Center for Computational Genomics and Bioinformatics. What that meant in terms of my scientific background was that I was a system administrator. I thought that I was going to be doing some kind of recognition code, some kind of interesting development, but the reality was I was working for a biology group. And I could either support the biology directly, or I could go, maintain the machines. So I started taking free classes, free tuition, got up on biology a little bit, and then joined this company called BioTeam. It's a tiny, tiny consulting company in this area, and we say that we enable science. And that's as corporate as I'm going to get. So Mark knew that that was my background, and he knew that we had this group of AI, A-life, artist people, and he knew that I get all geeked about this stuff. And so he suggested to Brian that I talk. Here I am. First off, words. I mentioned communication being difficult. You may have heard of bioinformatics, biology, proteomics, all of these new hot phrases. The suffixes are as follows. Ology is the study of. Ix is also the study of. An ohm, if you have a set of things, if I have cubes, the cubome is the set of all the cubes. The genome is the set of all the genes. The proteome is the set of all the proteins. Chromosome is just a colored body on a slide that's there to mislead you. It's noise. But somebody comes out and says, I'm studying transcriptomics. You figure out what a transcript is, you can know what the transcriptome is. And then omics is the study of the set of all of something. So proteomics, genomics, transcriptomics, economics, I don't know. The core problem, if you're going to be a computational person and wander into science, is that with physics it's pretty easy, because physicists fundamentally have been using computers as long as computers have been around. Most of them know Fortran, most of them have worked on a cluster computer, some of them like to talk about their days at the national labs, whatever. They probably want root. They are probably going to come in with some hunk of code and say, this needs to go faster. Mark is one of these. He walks up to the cluster and says, I have this awesome piece of art, and it's implemented in C like this. Love that. Biologists, right now, to get an undergrad degree in biology, you do not need to have any computing course whatsoever. Most universities. Some of the tier 1 universities are shocking innovations, stuffing one programming course in for a science degree. I predict that that will continue and that will happen more and more. Right now, a lot of practicing physicians, nobody claim that physicians are dumb, that they don't know their science. No programming whatsoever. And yet, what I'm going to tell you is that in, say, this year, this calendar year, physicians are going to have direct clinical level access to whole genome level of information. So 3.6 billion letters, your genome, 30,000 genes, your physician will have access to it. And they will have, I predict, very, very few of them will have any idea what to do with it. It's an interesting challenge, and one that is almost purely communication, honestly. So more about words. If you're a biologist, a matrix is this gel thing that particles move through. A vector is either a mode of infection or something that you use to get genes into things. Well, words overlap a lot. So anyway, this was my first machine room. It turns out that if you're going to deal with biology, what you need a lot of space for is data. And the reason for that is that biologists have managed to automate a tremendous number of their experiments. Back in the bad old days, you would have Bunsen burners and pipettes and whatever, and do an experiment, get to the end point of the thing, and have a result. Go to the library, think about it, go to conferences, maybe publish on it. At this point, that process of moving fluids around and heating them and cooling them and having biological operations happens to 384 well plates that are about yay big. So you do 384 instances of the same experiment at a time, and actually a robot is running them. So literally 10,000 of this experiment in a day. And so previously, one experiment, think, read, talk. Now 10,000, stuff it on the RAID. Another 10,000, stuff it on the RAID. And so all of a sudden, in basements all over universities, all over the country, there are piles of compute infrastructure, piles of disks, piles of stuff accumulating. This is why it's very, very easy, if you know a little bit about systems and a little bit about programming, to jump into a biology group and say, hey, I can help. But that's why you get stuffed in the basements until you learn biology. So these days, I deal with systems more like this. This is actually two 60 terabyte disk arrays. This is one 60 terabyte disk array, and there's another one back here. And then there's a 50 node compute cluster in the middle, all maintained by less than one system administrator, which is why they hire us every now and then. They've suffered 2 direct lightning strikes to their building this year. I don't want to work with them anymore, they're unlucky. But this is actually not even biology, this is NASA. This is, they call it the Calypso project, satellites capturing cloud climatology data. It goes onto these disks. And that's why I get all excited about this stuff. Because, you know, these guys are putting up papers about global warming, all kinds of good stuff. Anyway, this is what I find frequently when you come into a basement where there's not sort of a dedicated core IT team that's built up over 50 years. Somebody said, hey, I'm the NIH. Here's $10 million. Start your compute infrastructure. And you have a classroom. Well, then extension cords go out under a door. And that's actually a really bad idea, because energy comes in fairly cool on these cords, but it gets turned into heat by the computers. In this particular case, they overheated the room, shut it down, blew out the fuses on the floor, shut it down, blew out the fuses on the building. Had to negotiate with the janitorial staff to not buff the floors while they were running their benchmarks. That's a good time. This was their network infrastructure. They actually made the top 500 list, though.
They wonder why they got hit by lightning?
This is actually a different one. This is not NASA anymore. This is a university in Maryland. But yeah, good times. There's lots of good work to be done. So anyway, just so we can be on kind of sort of the same page with the biology, I'm going to blow through way too fast what I think of as getting up to the sequence level of genomics. And I apologize, it's going to be too much data, nothing I can do about that. It's a professor I like to work with, Nevin Young, University of Minnesota. He says, bioinformatics is full of pitfalls for those who look for patterns or make predictions without a thorough understanding of where biological data comes from and what it means. The reason I share that is that the world is replete with stories of mathematicians, physicists, scientists wandering up to biology and saying, God, this is simple. You have this dead simple thing. I've got a nonlinear curve fit algorithm, be it neural nets, be it whatever. I'm just going to solve this for you. My favorite story comes from actually Minnesota. We had a visiting physicist on a sabbatical from England. He came in and said, show me your hardest problem. I'm a smart man. Show me your most difficult problem. He said, well, you know, that's really kind of fuzzy. You know, there's a lot of stuff going on here. He said, no, Bob, I'm smart. He said, okay, gene finding. You've got large chromosomes. They're just letters. You can just imagine that for the moment. They're just long strings of letters, 3.6 billion letters. Find the genes. We've got some of them. We'd like to know all of them. We're going to hide half of them. Why don't you find them? And he comes back, a couple weeks later, says, your data is crap. Your data is absolute junk. I said, well, yes, but how did you know? He said, well, half of your training set is not even in this data. You told me the gene was here, and I looked there, and it's something different. It turns out that DNA runs on a forward strand and a reverse strand, and sometimes genes read forward and sometimes they read reverse. It's about 50-50. He didn't know that, and had wandered off and spent a couple weeks wandering through his algorithms, and so it's very, very easy to waste a lot of time on this stuff. It's very, very easy to download 20 gigs of data, sequence data, and try and dig through it and find a good pattern. And what you may find is a well-known source of instrument error. You know, you may find a well-known thing. So find a biologist to work with is my advice. What is bioinformatics? First time, I'll say this 3 times, different answer every time. Bioinformatics is biology, and you just have to kind of deal with it. There's this social thing where if you get a PhD and you wander into a room, you want to think that you're the smart person. And if the biologist also has a PhD and walks into the room, they want to think they're the smart person. And the dark reality is that you're doing that biology. That's why I ended up in the systems room. Anyway, all the way back, 100 and some years ago, Gregor Mendel wanted to be a math professor, didn't pass the test, wound up a monk in his garden, did some amazing experiments with peas, reproducing peas, you know, and he came up with the fact that traits seem to be passed by a combination of factors in the parents onto the children. And he did some remarkable math, really, to say, okay, I've got these distributions of 25, 50, 25 in some traits. And I can explain that if there's something going on in the cells that I can't see. But it's a mathematical construct somewhere in there. You know, he tossed it to God, he was a monk. And it turns out you can get 3366 distributions if you say, if you've got the two of one type of gene, you see it. If you've got a mixed, you see it another trait. But if it's 2 of the other side, it dies. That's how you get 2/3, 1/3 versus a quarters. And it turns out if you've got those 2, you can get most of the interesting fractions. So he published in 1866. His primary journal ignored him. He had to push off in some dumb little journal. First cited several years after his death. Poor Mendel. But he was the first one to really come out with this idea that there's hidden traits that you can't see. There's a genotype, something about the genes. He called them factors in German. And then there's a phenotype, there's the physical aspect that you can see. And then in 1902, Walter Sutton said, hey, there's these things that kind of go in the cell. We'll call them chromosomes, colored bodies. And he looked at spreads like this and said, you know, it kind of looks like there's long bands that are crossing over in the middle and might be like pairs of factors interacting. You know, not at all obvious. He was working on grasshoppers. He got lucky. Grasshoppers have freakishly huge chromosomes. And then finally, Thomas Morgan, you know, a few years later, was working on fruit flies. Why fruit flies, you may ask? They're cheap, they're small. His grad students bought bananas once a week, you know, and his grad students were looking at whether the eyes on the fruit fly were red, tracking the sex life of a fruit fly. And there was this guy, Alfred Sturtevant, who was an undergrad, who was in there moving fruit flies around and feeding them bananas, and he had algebra homework to do, and he didn't feel like doing it. So he noticed, everybody had noticed, that nobody could quite replicate Mendel's precision in his math. If you look at Mendel's paper, the numbers are perfect. There's no decimals. It's like 25%, 50%, 25% every single time. Well, in reality, it's not that clean, and everybody thought it was just error, you know, or maybe he was just cleaning up his data. Sturtevant said, well, what if I just say that the degree to which genes seem to hang out together, maybe that's not just error. What if I try and arrange them in a line and say, more linkage, more they hang together in the reproductive process, closer together on the line. It turned out he used his algebra that he was not doing his homework for, and made the first genetic map of Drosophila. You know, this is everybody's dream. He then eventually went, I think they let him go to the Nobel ceremony, but he didn't actually win the prize. And that's the picture that he made. So if you're laying things out, these factors are now being associated with a chromosome, a position on a linear molecule. You've now got sort of a physiologic trait that's somehow linked onto a place on something in the cell. This hasn't been obvious for long. I mean, we're talking about one human lifetime that we have been able to say this sort of thing. And then everybody knows these guys. Watson and Crick said, hey, there's this crystalline DNA stuff. Maybe it's not just repeating polymers of GATC, GATC. It was referred to a couple years prior to their publication as a stupid tetranucleotide. They said, hey, if the order was allowed to flex around, it could be a code. You could like put information in there. And, yeah. And Watson proposed something that he called the central dogma. I think more of us should have dogma. I think we should just admit that, you know, he came right out and said, I have no reason to say this other than that I know it to be true. You know, that everything that we have in DNA, we have all the information we need that gets transcribed into some messenger molecule and translated into proteins, and you build your organism out of proteins. So this is stored program computing. It really is. You've got something stored, transcribed, translated, goes out and makes proteins. And so in the 60s they decoded the genetic code, figured it out. This is another instance where there's I forget his name, well-known physicist, who wandered up and found a very beautiful code that would work and not have any overlaps, and it was totally unambiguous, and you didn't need start and stop codons. Totally wrong, very beautiful. It turns out the actual code is highly redundant and survives noise very, very well. It's not beautiful, but it works. So genome sequencing, and I'm coming to the part where we go into genomics here. Sanger in the 50s came up with this chemistry by which you could take a hunk of DNA, it's up at the top here, chunk it off so that you have substrings of every possible length. You're breaking off every possible endpoint, and then weigh them and have them sorted by weight. And that's fundamentally limited in a pretty obvious way, because the weight of one endpoint, one nucleotide, has to be significantly enough different from the weight of the strand that you're reading that you can still separate them. This is limited to about 1,000 base pairs, and that was true until 3 years ago. We were not able to read more than 1,000 letters at a time out of this 3.6 billion letter book. They got up to, 700 base pairs is what they sold, and remarkable stuff. So the human genome, when they read it, started in 1983 just laying out those chromosome maps for humans, for our chromosomes. It's been completed several times, mostly because you can say done is a number of different things. The interesting bit to me for how this differs from this year, when they finished this up, they read it in 1,000-letter pieces. They read a 3 billion letter dictionary in 1,000-letter pieces, which means about 3 million independent laboratory experiments to read every letter once. You don't read every letter once. You read it once forward, once backwards, really more like 5 times forward, 3 times backwards to be sure. And so this is just a vast amount of human effort to separate out, to move chemicals around, even with robots. If I'm doing 10,000 a day, that's a really long time. They resequenced Watson last month on new instruments, and they finished it in a month. So this is changing things. It's, yeah.
Just quick, there's a new X Prize on just
yes.
this one. And you've heard. Basically, they want to do it in the X Prize for, I believe, 10 people in 2 weeks.
Yeah.
For under a certain amount of budget, I think. That's also a requirement.
The funny thing about the X Prize for me is that the X Prize for space flight was necessary because you're not going to get private industry in without a whole lot of help. For genomics, I know somebody, they're like, we're going to have the $1,000 genome like this year. And it's going to happen because pharmaceutical companies want it, pharmacists want it, physicians want it, patients want it. I want to know if, my daughter has the breast cancer gene so I can start her on whatever when she's 3. But yeah, it's going to be remarkable. I'll just skip over that. So genomics, back in the bad old days, they usually use this as a parallel computing slide to say, okay, I built clusters, this is not a new idea. These were the spouses at the project with nuclear explosions and stuff. They put them, Manhattan, yeah. They came up with algorithms by which they could actually harness all that computational power. Similarly, if you change this, if they were doing biology, and you changed it to this with a row of machines and a couple tests, you can kind of see where the problem is going to come in. And this is if you take year along the bottom, if you take number of sequences in millions in the public repository, which is called GenBank, where if you get money from the US government to do genome sequencing, you have to deposit the sequences that you get into GenBank. And it's exponential. It was exponential in 2000. It's still exponential. You kind of have to show this slide, but I never update it. Next-gen sequencing, this is the fun part. This is where we kind of transition from things you might have heard in high school to things you probably didn't. This is an instrument called a GS20 from a company called 454 that was purchased by Roche. Costs $1 million to buy the instrument. You can read about 500 million base pairs in a single run, which costs you $7,000 of chemicals. Takes a couple days, generates 2 terabytes of raw data. And you can do that non-stop. So and these are the machines that they use to do a whole human genome of Watson in 30 days. This is the first edition of a new generation of sequencing machines. This is a different chemistry that is not fundamentally limited to that 1,000 base pair read. There's no reason in the world that we won't next year see these things just cranking out entire chromosomes. Or that you won't see one of these, or its children, its descendants, costing $50,000 at your local Dock in the Box. The question is Roche, and this is where we come into the things that I do on a day-to-day basis. Roche is a chemistry company. They are a robotic chemistry company. They're a diagnostic company. They don't want to be a computing company. They don't want to be a software company. And what they're doing is creating a data storage problem and a computational problem. It's kind of fun to watch. And the question of how you would take that from a research application, which is where it is now, to a full-on clinical application is going to be remarkable. And it's not going to wait. If you go online and look for a genomic paternity test, I thought I had a good idea recently. I was like, okay, I can do this. I'm going to do the full-on paternity test. I assume it'll be cheaper soon. It's already out there. There's like 5 companies that you spit in a cup, and if you have a kid, you have them spit in a cup. You know, it's got a little checkbox. Is this mom or dad or kid or whatever? To summarize that, most of the diagnostic stuff that we do is primitive right now. If you look at the reaction to the anthrax scare at the Capitol, they got this white powder, and then they sent it off and literally were like dipping pieces of paper in it to see if it turned pink or blue, you know, and heating it up to see when it melted, and putting it on bread to see how rapidly it grew, and graph paper and everything. Where if you have the complete genome of that beast, which I just told you, I didn't say it out loud, but I just told you can do that in a day now. You can compare it against all the other ones that you've got in your library, and you have the answer. That's another fun project we're working on right now, is a DoD facility where basically you get the white powder, turn around in a day, and they go shoot the right people. It's kind of disturbing that it's that easy, it's that dead simple. There's no question to it anymore. Not like, you know, the DNA thing with OJ or whatever criminal. You know, sure, it's 1 in a million, it's 1 in a billion. It is the entire genome. That's the transition we're looking at. So I mentioned scientists in basements, computers in basements, computers in closets. Give a biologist the ability to generate 2 terabytes of data a day, give them NSF funding, and they will. There's a fascinating, fascinating conversation going on right now about, so lab scientists are terrified of ever throwing away primary data. If you do an experiment, that's really hard and really, really expensive. You never ever want to throw away your primary data. And yet, if it's 2 terabytes a day that you're cranking out, I don't know, you know, do the math, but that is substantially more expensive than $7,000 for a run if you're going to keep that data forever. And so they're already starting to have this conversation about, hey, if we've got the stuff in a freezer, if we sequenced a genome, if we sequenced a person, if we've got a pathology sample, keep it in the freezer. That's like electricity to keep it cold. And then we can just throw it through the machine if we ever actually need that data again.
You do need to have some fingerprint form of it if you're going to do that. You know, what is this one power?
That's exactly that's exactly it. 2 terabytes is way, way more than 500 million letters. And so what they're stating out here are images that they're taking with high resolution CCDs. And if you keep the images, it's 2 terabytes. If you keep the base calls, the letters, you know, it's a couple megs. And they're very, very hesitant to throw away the images, but they will absolutely have to. You know, try convincing an astronomer that they'll have to throw away their telescopic imagery. It's not going to happen. And yet, it has to happen. And I mentioned NCBI as this repository, well, maybe I didn't mention NCBI. The GenBank is hosted at the National Center for Bioinformatics Information. I have no idea what they're going to do when FedEx packages of disks start arriving. Like, previously you could just upload your data, and it was required. In fact, they demand that this happen. You upload your primary data, and it be stored and publicly available for everybody. First off, how do I send 2 terabytes a day? That has to be a FedEx question right now. And second, what's NCBI going to do? Put them on pallets in the loading dock?
Well, are they requiring the pictures or just the code?
Right now, it's one of these things where the NIH's legalese documents are not sufficiently clear. And so some people think that you actually need to push in the instrument, the images. So terabyte scale data. Here's something fun. Back out of data overload, if you wanted to say something fun about genomic data, you might say, I've got a sequence. It's this blue bar here. It turns out that it's in some piece I was reading chunks of the sequence. These are the chunks that I'm reading along some larger chromosome scale, and I'm hanging off information about similarity. There has to be a better way to represent that visually. It was very confusing to me at first, but there's a ton of information here. And honestly, this is the best that biologists have, at least as far as I know, in terms of saying, I've got a chromosome up top, I've got a piece of it, I have some sort of visual context for where I'm looking at. and I'm hanging information annotation off of it. No, big visualization question. But I just wandered through sequence. I took you all the way from Mendel up to genomic level sequences, chromosome sets of letters. Turns out that is only the very first level, and I mentioned in passing this central dogma where you take DNA, turn it into RNA at some rate, which you're controlling, and then you turn that into protein and that builds an organism. And if you want to talk about drug metabolism or something, you know, drug comes in, turn on a gene, things start happening. That's all the way down at a level where there are massive levels of interaction between these things. People really, they will say they know what's going on, but it's difficult. Microarrays are fun. Here's a fun bit. You can figure out which genes are being expressed at any given time. So this is the spit in a cup thing. Spit in a cup, and I can tell you of your 30,000 genes, for about $700, which genes are turned on and which are off right now. That seems like an interesting question. The problem is that it's still an image processing question at some level. You've got these spots, and this is literally fluorescing molecules that are in these little itty-bitty circles, and the question is, when is it on, when is it off? You're talking about solutes being washed over a plate. Very, very challenging, but it's something else that you can do. And you never get a really clear answer to what a gene is. You know, is a gene the fundamental unit of genetic inheritance? Well, that's what my genetics professor told me. He also, when I asked about bioinformatics and if we were going to cover it, he said there is a word for the application of mathematical and programmatic models to genetic inheritance. It is genetics. You know, if you write a program to do genetics, he didn't care. I mentioned these SNP chips. This is something that's really fun, and I'm coming to a conclusion here, and I'm just going to start throwing out sort of things that I think are cool. SNPedia. I mentioned that there are about 30,000 genes in humans, give or take, whatever a gene is. A friend of mine actually downloaded that list of identifiers for all the 30,000 SNPs. A SNP is a single nucleotide polymorphism. That's the closest we come to a gene. So if you have one letter that is different in some humans from others, these are the points where humans vary. We know pretty well what about 1% of them do. You can get all of them right now. You can figure out your complete genotype. It's about a $1,500 question. You go to SNPedia, you can find the companies that'll do it for you. So all he did was he pulled down all the identifiers for all of those genes. And he provided an import thing so that you could say, I got one of these reads from one of these companies. I would like to know if there's anything that anybody has left in this wiki about my genotype. And you go to your, and then that wasn't very interesting because not many people were getting it done. So we went to PubMed, which is this repository of biological publications. Said if I find an identifier for this gene that I already have in here, take the abstract and stuff it in the page for that gene. Nothing clever, nothing smart. And then people started sending an email. Then people started really using this thing. And then he did something that I thought was just ridiculous. He turned on chat rooms. So if there are multiple people looking at the same genotype, interested in the same area, he allows them to interact and ask the question, okay, it's an unannotated gene. Nobody knows what it does, but we both have it. What do we have in common? It's the least scientific study you could possibly do, you know, in terms of self-selection, but big piles of data, what are you going to do with it? You know, similarly.
Can I get you back up a second?
So sure.
You said, whatever it was, $1,500 you can get this tested. So that tells you that for every one of the points of known variability for humans, they'll tell you which you got.
Exactly. If we know that humans come in lowercase a and uppercase a for each of 30,000 instances, it'll tell you which you have. And for some of them, it's like bing bang breast cancer. You know, dead simple.
So that's an easier test, a cheaper test than reading the whole thing.
Yes.
Just, they've got specific markers they look for.
And it could be, you could claim that it's the useful 30,000 points. And the companies that are selling this do make that claim. And it's also a fact that there's all kinds of other gunk in there that is not being measured by that. But it's what we can do today. It's what next year some pharmacies will probably have the ability to do something interesting with that information. Where one of the genes that we know pretty well is the rate of metabolism for a particular serotonin reuptake inhibitor. That's your mood drug. So it turns out you're either slow or you're fast on metabolizing this thing. So you probably either need more of it or less of it to have exactly the same effect. If you told your pharmacy that bit of your genotype, they could more accurately dose you. Gee, that seems like a good idea. That's like next year. Something that I think is fun is that biologists have been traveling the world for 500 years, separating out bears and looking at length of arm and joint location and tail and fuzziness of ears and whatever. And they have this idea of a species and the idea of a phylogenetic tree. You can do it genetically as well, and the answers come out mostly the same, but with some surprises. I'm just shocked by the overall how right these biologists got it when they were like measuring foot length. And you look at the DNA, it's like, yep, that's how it evolved. I'll just skim ahead to the thing that I thought was the coolest. I was at Supercomputing last year, and the Blue Brain Project. Has anybody else heard of this thing? But I think it's amazing. You can simulate a single neuron down to the molecular level, you know, and you can simulate many neurons, and then you run out of CPUs. But if you've got a Blue Gene machine from IBM, you can do a really big simulation. Their goal is to do a complete simulation to the molecular level of a neocortical column, which would be sufficient to drive around a small insect. And then they're just going to hitch themselves to Moore's law and ride that up to a full human brain simulation. And I think that that's amazingly cool. I'm sure it's somehow bioinformatics too, but I don't get to play with them. I just see them at conferences. And I love their little hedge at the bottom here. We don't know if consciousness arises because of some critical mass, blah, blah.
So where did they get, did they just start with basics about neural networks, or did they scan at great accuracy and actual neurological slice?
They've never done any neural networks.
What's that?
They're not doing any neural networks. They're doing molecular level simulation of actual nerves.
Right, they call it a structure in the brain. Where did they get that? Do they have an extremely accurate slice?
Yes. They started off with C. elegans, which is this little nematode worm.
Yeah.
That, I forget how many cells it has, but we know each and every one of them and what it connects to.
Right.
They started with that connectivity and started working up experimentally observing, building it in, experimentally observing and building it in.
I mean, is all the neurological processing done so well? Aren't there still mysteries about how things communicate between neurons, or is it all done so well that you can simulate everything? I mean, I know that they simulate C. elegans. Does it behave just like real C. elegans? I mean, they verify as they go along.
The trick with simulating C. elegans is simulating its environment.
Well, you can
provide
C. elegans in a simplified environment and have you simplified simulating C. elegans and see if those.
Right. And I
think that to simulate the entire garden.
Right.
C. elegans happily.
I'm not sure where they stopped with that. I'm sure that they did some basic validation because they talked about it at the conference thing, but I honestly have no idea.
Have you read stuff that Kurzweil wrote?
Yeah.
That would seem to be a direct tie. Kurzweil was.
Did you see him when he came to MIT?
No,
he
I missed it.
that
He
debate.
was certainly saying that eventually they'll be able to scan three-dimensional entire brain and then essentially capture them in a computer and transfer your intelligence to the computer.
Yeah, which leads you to the Star Trek question. You know, if Scotty comes up to you and says, well, you know, I'm sorry, the transporter worked, but it didn't blow you apart at the end, you're actually on the planet, but I have to kill you now. Regulations say so. Like, wait, no. Yeah, that's actually my Kurzweil slide is the observation that if you, this is a top 500 thing, if you take the sum of the top 500 supercomputers in the world in terms of teraflop performance and plot them out over years, it's a nice straight line on a log scale. And if you take the top 500 that's kind of bouncing up in these little steps as a new regime takes over, what's really, really fun to me is that this is my laptop. And it tracks right on along. You know, so all you have to do is wait 4 or 5 years and you have on a laptop what it's possible to do with the biggest computer in the world right now. And I think that that will actually save us with regard to these clinical questions of genomics, where 2 terabytes seems like a lot right now. It's going to be in my pocket, in my USB fob earring, you know. So, what I said, bioinformatics is biology. The underlying science is nasty and hairy, and if you don't get into that and chase it as fast as you can, then the computational results that you derive are going to be suspect. And if you're flexible enough to be able to walk into a room and constantly not know what's going on, there's tons of good work to be done here. And there's a bunch of other stuff too. I just threw words out there to scare people. That's what I got prepared. thanks for the opportunity.
So you kind of touched on this one. I'm just curious in terms of, so now that we're going to have, on the high end, more and more, more and more flops that are going to be able to be used, what do you see at the high end as things that will happen in the future? So as I know, kind of under Dwan, we'll have pharmacies doing all these things in terms of, besides the consciousness simulator.
At the high end, we're going to actually have small organisms simulated down to the molecular level. I don't know whether that's 10 years out or 50 years out, but it's going to happen. Right now, there's a lot of, they call it metagenomic information, where we say the central dogma, and it's DNA makes RNA makes protein. But it is also the case that a fruit fly mom, when she creates an egg cell, makes a chemical gradient in that cell so that one end has a whole bunch of one chemical and one end does not. The end with the chemicals on it becomes the head, the other end becomes the tail. That's not genetic information, that's something else. And I think that we're going to start getting enough of a handle on the genetics and the genomics to be able to say, just like Sturdivant did, okay, there's noise in this, it's not working. We thought it was just a reading error, and we're going to notice a whole new domain of biology, sort of in the gap there. I think that's where the big guys are going to go. All right.
Where do you work and what does your lab do?
I work with a tiny, tiny company called the BioTeam. There are 7 of us. We are all scientists from one background or another. We've got a biologist, a physicist, and we're all into high-performance computing. And so we've made a nice little niche market of wandering up to these smaller labs, and in fact we've gotten up to a Fortune 20 company, and saying, look, there's a big communication problem between central IT and research scientist. And you need somebody who can speak both languages. Go in for a week, go in for a month, and have fun, talk to the mad scientists, talk to the IT folks, solve some problems, make recommendations, and move on to the next thing. We also have a hunk of software called Inquiry that Apple sells. It takes some open source tools and dumps it on an Apple.
So actually, another question is about cost. You said that to do the run to map the entire genome, is it 7,000?
So
for one run for the chemicals?
For one run for the chemicals is 7,000 right now, and honestly it's because they use the HP scheme, where they give you the printer below cost but then charge you for the ink.
Right, okay.
The problem with that is that it really, really has offended some of the folks in the community who came up with the chemistry. And so they're coming up with slight one-off chemistry. You know, the actual cost of these materials is not $7,000.
So I mean, just the question I have on it, in terms of the storage question, you say all you want is a raw copy. You can get for like, I think about $225, you can get a 750 gig hard drive. So if you wanted to get enough to store the data redundantly, say all you're doing was a really sucky job, but if you just put the drives in there, load it to them, and then set up the rig, and then took them out again, shoved them in the closet. But still, so now you're adding somewhere below $2,000 to the cost of the run, which is doable. But it's just that if you want to actually use that, then it's going to be other. But if you just want to keep it, keep it just for the hard drives.
I heard the term MADE at a conference, a massive array of idle disks, which is what we're building. And there's the Sanger Center has this model. They can't get out of keeping all the data, so they bring in a rack of storage, and they fill it up, and then they unplug it, and they move on. And as soon as anybody ever asks for the data, they promise they'll plug it back in. And it turns out that when you need the data is while you're actually working on it, while the publication is coming out, while you're doing that project, which means you're probably living in that same rack. And you've got like 2 of them going at once. And then a month passes and nobody's asking.
So once you have all this data, how is the software for actually looking through it and discovering what's there and finding which genes you're looking for and that kind of analysis?
It turns out that the standards for that are crap. The questions as dumb as file formats are still plaguing us. It doesn't help that we're dealing now with diagnostic instrument companies who make a lot of money being one platform silos. You know, there are XML standards, there are metadata experimental standards, but in terms of just the raw tools for sifting through, that was the reason I mentioned Snipedia. Is honestly, this was the first time I've seen something come out. It's like you've got 30,000 genes, you've got the ability to do this experiment. Why not just toss it in a wiki and let the collective consciousness deal with it? It's all custom. Most of it's written in Perl, unless you're in a big pharmaceutical house, in which case they have paid lots and lots of money to have their special thing. But then if you get fired by Novartis and you hire in Bristol Myers Squibb, you have to learn a whole new platform. So it's bad.
You mentioned that there's this human being will be sequencing or creating a simulation for small biological organisms that get more and more complex, and to the point of maybe reaching consciousness. Right? You're talking about that in your slides there. Does this imply that our own consciousness is just a trick of the biological machine?
Do I need another here? That's religion.
Well, I mean, is that sort of the premise?
Well, all you can ever do with science is measure observations, observable things. And if you can tell me how to measure from the outside consciousness, then I can come up with an experiment that will answer that. And if you can't, then we're just going to have to sit around and drink.
Right.
You mentioned that there's a built-in premise that there'll come a point where, at the cost of a Band-Aid or something, you're going to be able to sequence your own genome. But it's not entirely clear to me that people really want this. Because you mentioned that if you had maybe you did do it, if you had a 3-year-old daughter, and I'm not exactly sure you would want to know the fact that she's at risk for breast cancer. If somebody would have scanned me and told me that there's risk for cancer or there's risk for Alzheimer's, these are incurable diseases, so there's no therapeutic approach to dealing with these. My reaction would be that you would not want to know, because it just essentially ruins your life.
The best example of that is probably Hodgkin's disease, where sometime between 35 and 40 years of age, you become senile.
Yeah.
It's really nasty.
Sure.
Totally 100% genetically determined. And you can just tell. You can get this test now.
You know it, maybe you should know when you're your teens or your 20s.
However, the trick is that there are going to be so many advantages, and this is my speculation. There's going to be so many advantages to going to the pharmacy and getting the right dosage. These just that simple things that we won't be able to avoid getting that information, and we're going to have to somehow deal with that bag. Like the problem with these companies that do the SNP checks, where you can get your 30,000 genes. Informed consent is meaningless when you don't know what 99% of the data does, and it's going to come out during your lifetime. If I give you, if we can test you for Hodgkin's and not test you for all these other things, but if it's actually cheaper and more effective and standardized to do it with this one test, and we're going to do it to all the, and I'm just thinking, 15 years down the road, it's going to be like blood type.
But with the HIPAA regulations, et cetera, we might say, I want you to do the whole test, use the one part needed today, and throw everything else out. Because I don't want my insurance company to go and get it. Decide I'm uninsurable.
I think maybe the most fun thing that I've come up with for that is the concept of race and the concept of culture. Where right now we have some really simple heuristics for deciding if somebody's in group or out group. Language, hair color, height, skin color, whatever. I think that when, I don't know if it's 15 or 30 years from now, but kids are going to have USB fob earrings that'll glow red if you're related within 7 generations. I think that's going to play hell with the concept of culture.
Yeah, that was actually one of the things. Several states, Massachusetts is one of them, I forget how many, have introduced where in the discrimination clause we're not allowed to discriminate with sex, race, religious preference, or genetic information. So that's actually in the Massachusetts code right now. I think most states don't have it in yet, but that's pretty good thinking for the future. Because what I'm thinking is that this is going to be hell for the health insurance company. Talk about pre-existing risk.
Yeah.
I mean, on the one hand, it's good for them to know what, because they'll be able to better evaluate your insurance, like how much risk they're taking on when they're taking you. On the other hand, so if you're sick, they're going to know not to take you in. But if you're healthy, you're not going to want any insurance. I mean, you take this test, it says you're completely clean. don't get the flu. That's true, but instead of paying like every month for health insurance, I mean, you pay for ER once, and it's still way cheaper. So, sort of clean bill of health, you're not going to want this. So health insurance companies are really going to be against this. So it's this really
Clearly haven't had a ER bill.
right. Well,
Does your genetics tell you whether you're gonna, trip and fall into the street and get hit by a bus?
that's so you're not going to want to pay as much as you are paying now. So because I'm probably not going to go in for sort of long-term care for anything other than some sort of physical injury that's going to mess with their current practices and their current policies and so forth.
Interesting argument for universal health insurance. Everybody just paying because, as a society, a certain number of people are going to die every year.
I just on one note on the Alzheimer's, I think that if we live longer, we fairly well agree that everyone will develop a form of Alzheimer's that will like dementia. And so what I think might happen is that there's one company that's produced a chemical that emulates what's in fish called acetylcholine. And the acetylcholine that's produced, whether it's more, not produced, but stopped from being washed away. So once the acetylcholine is in the ceramics, it doesn't go away. It's there. So what you're going to have is that more motivation for people to invest money into companies like Roche or whatever that want to develop a better version of that, so that in effect, knowing that 30 years ahead of time, 10 years ahead of time, will result in more money being poured into a better version of what we already have now for Alzheimer's.
You're saying a young rich person finds out he won't have Alzheimer's
Or a
in
parent,
years.
or an investor, or whatever. I mean, the investor is going to make money anyway. The company's going to make money off it, and the person who is going to develop a problem in their life is going to have a better version of a fix than we already have. Because we already have a really, sand up program, for people to fix it. It doesn't fix it, it just stops it from getting worse fast.
So like the Moore's law of
Can I
solving
get
problems.
Yeah, wait, really.
So I have a question before we talk about things that would simulate neurons, and then you could go from that and simulate brain. It seems to me that wouldn't it be possible to, well, I have two questions. First of all, is that do you mean that later on down the road we'll be able to do that real time, so theoretically you could have conversations with the computer, or do you mean that later on down the road we'll be able to because it seems to me that right now the only power will be processing power, or maybe also memory, but mainly processing power. So in theory you could compute for a whole human brain, it's just that it would take a long time to do it. So
They're doing it real time.
oh wow.
It's going to be sweet.
And my second question,
Yeah.
my second question was, you said something about simulating at a molecular level, and that seems to me sort of like simulating a computer where it says simulating the logic gate, you simulate each atom. What do you mean molecular level? Wouldn't it be simply?
The reason they made that decision is because we actually don't know exactly what all the dynamics of neuroanatomy are. We know most of them. Most of them have been observed. But if there is something that's really, really critical that would turn out to prevent consciousness from ever arising, whatever that might mean, they don't want to miss it because they built in their model ahead of time. They want to go all the way down to molecules bouncing around and Newtonian forces and build up from there, so that if there are other factors going on, they'll just be built in from the start.
So I imagine they're working with, simulations of molecular level simulations of things like neurotransmitters, but obviously there's a lot of things going on inside of the neuron that
they believe
get to more fundamental problems that we still don't know about and still have to solve. So aside from just the memory and the processor limitations, we have the understanding limitations, which is also related to why we have so much data that we don't want to throw away because we don't even know what to do with it yet. So I think there's still sort of a long horizon of figuring out how that stuff works before we can really use it.
Actually, at the level of simulating the neuron, there's a recent set of 3 papers published on Archive about the transmission of the neural impulse, which strongly suggests that Hodgkin and Huxley were wrong. It's absolutely fascinating series of papers. It makes a lot of